Hey everybody, hi, welcome back to Lockdown Learning. It is lesson number 32, the last one of this week. It's a public holiday in the UK tomorrow, so there'll be no lesson tomorrow, but we're here today, and as promised at the end of last week, we are getting round today to determining differentially expressed genes. So we're going to use the functions provided to us by DESeq in R for finding genes that are differentially expressed in our now 2 datasets after we split our datasets up yesterday because of our initial PCA plot. So if you remember yesterday's lesson, so yesterday the aim was to try and look at PCA as much as possible anyway. And the reason for that is PCA being a very effective technique for assessing the similarity between samples in an RNA sequencing experiment or in lots of high throughput techniques. And our initial PCA, which you can see here, the initial PCA was really demonstrated that what we're dealing with here is 2 experiments, not 1 experiment. So we split our data set up and then looked at the principal components of each of those other experiments individually, and they look much more like real, quote unquote, real RNA-Seq experiments. These look the way that I would expect an RNA-Seq experiment to look. Essentially, is what I mean by that. And then we looked at some other ways of looking at. We looked at some other ways of assessing the similarity between samples in an RNA-Seq experiment. So we looked at hierarchical clustering. We did this quite quickly at the end, what I will do, I think, once we've got some differentially expressed genes, and we've dealt with annotating those differentially expressed genes, which is kind of the task that we'll undertake in the early part of next week. We'll come back to these techniques and look at them in a bit more detail, because as with all these things, I am sort of aware. I do have some awareness anyway, and I do sometimes realise that I go over things quite quickly, especially as things have got more complicated and more detailed, that sometimes they can go by quite quickly. So we will come back to this. Okay, so this was the hierarchical clustering. So we found the distance between our samples using the dist function, which gave us a distance matrix, and then we applied hierarchical clustering to that distance matrix to give us this dendrogram. And then we also looked briefly at supervised clustering. So k-means clustering was the technique that we looked at. We split our data into 2 clusters and saw that it divided exactly how we expected it to divide. That the three samples in cluster 1 were 1 condition, and 3 samples in cluster 2 were the second condition. So the data split up in the way that we expected it to split up. And so this is all encouraging in terms of the quality of this as an experiment. Now that we've subsetted out to 2 separate experiments, we can be relatively confident that the experiment looks the way that we expect it to look, and that I think we will be finding some differentially expressed genes when we get to that bit of the analysis. So what I wanted to do today then was to get through the remainder of that analysis. So to get from this point that we're at now, where we've done a certain amount of kind of assessment of the quality of the experiment. We've got 2 more steps in DESeq to apply. So we've previously estimated the size factors of our experiments, and we've got 2 other steps to take with DESeq to estimate the variance of our experiments. So there's a function to do that, and then to apply some statistics. And then we're going to extract results. So we're gonna pull our differentially expressed genes out of our DESeq dataset then at the end. So that's the process for today. So DESeq, the way that DESeq2 does analysis is it's a 3-step process. So 3 steps to DESeq analysis. So the first step, which we've already done, is to estimate the dispersion. No, sorry, take it back. Estimate the size factors, which we did for the first time a couple of days ago now, and that is essentially a normalization process. So that is the process that estimates these normalization factors by which we can then apply those factors to our data, and that allows us to normalize our RNA-Seq data, which is very important because we need our RNA-Seq data to be comparable. So often what you find is you get systematic differences between sequencing libraries, and those systematic differences are due to the way that the experiments are done. So just looking for, here we are, the appropriate thing to show you on my screen. So when you do an RNA sequencing experiment, and I'll do a bit of an overview here, just while I'm waiting for my whiteboard to load. We're familiar with this experience. Taking longer than expected. It always takes longer than expected, so therefore I think it just is expected. Okay, still loading. We'll get there eventually, promise. Okay, it's to do with this picture that I've drawn. So I've tried several times to draw a schematic of a flow cell, of a sequencing flow cell, and every time I try to do that, I always end up not being able to use my whiteboard for some reason. It's extremely frustrating. It's loaded, hooray! But of course now my surface is disconnected from the internet, so you can't see the things that I'm actually drawing. Right, here we go. Let's give this a try. I'll stop talking. Oh no, I won't stop talking, that would be bad. I'll stop just waffling and I'll actually show you something. Right, so here's my whiteboard, and we've seen this diagram now a few times, and I'm not going to, for now, I'm not going to delete it. But this is the diagram that I wanted to draw. So this thing that I've drawn here, this kind of abstract picture, this is my diagram of a NeXT-seq flow cell. So this is what a NeXT-seq flow cell looks like. This RNA-Seq experiment has been done on a NeXT-seq flow cell. I have one of these on my desk at the office, which obviously I'm not going to be able to show you because I've not been in the office for 8 weeks. But this is what a NextSeq flow cell looks like, and the idea with these flow cells, and the thing that I've been trying to illustrate for weeks now, is that you have these 4 strips that go vertically down the flow cell. These are the lanes. These are your sequencing lanes. And on a HiSeq instrument, where you had 8 lanes on a flow cell, the lanes were all individually addressable, so you could load different things on each lane. The NextSeq has a single load point, and all of the libraries get loaded from that single load point. So you load your flow cell from that single point, and the microdix takes your libraries into your 4 lanes. So whatever you load at the top ends up on all 4 lanes, a mixture of it. So what you have to do to load your flow cell is to make a mixture of all of your sequencing libraries. So you prepare all of your sequencing libraries, and then at some point during the library prep, they get mixed together and then loaded onto the flow cell all together, all in one go. And so then you generate your reads. So from high output NextSeq flow cell, I think you get about 400 million clusters, so about 400 million reads, single end reads. So we're dealing with single end reads here, and those reads are divided up between the libraries that you load in this loading point. So you pool of your libraries, you put them onto your flow cell, you generate your reads, and those reads are split between your libraries, and you can assign them out to the different libraries by the barcodes that are attached to them. That process is to do with the way that works is to do with the molarity of all of your libraries in the mixture that you load onto your flow cell, which means that if you've got 20 libraries in your mixture that you're loading onto the flow cell, it doesn't mean that each one of those libraries will just magically get 20 million reads. You're not just going to divide your 400 million reads by 20 and each gets 20 million reads. That isn't the way that works. It's to do with the molarity in the library mixture that you load. So if you have twice as much of DNA from one library as you do from another, then you'll get twice as many reads from that library. So what you'll end up with is, in these 20 libraries, they may on average have 20 million reads each, but they will scatter around. So you will get some libraries will have less, some libraries will have more. So you might have libraries with 12 million reads, you might have libraries with 24 million reads, you might have libraries with 36 million reads, and so on, and any number in between. And this library depth is the thing that really is that's the biggest influence then on the sequencing depth at any given gene then. So if we've got a gene that we've sequenced and we have reads that align to that gene in our experiment. So here's my aligning reads that come from fragments of mRNA that have been converted to cDNA and then sequenced. In a library with if this library has 1, 2, 4, 6, 7, I don't know, 12, 13, 14. So 14 reads mapping here. If the underlying sequencing depth here is 12 million reads, and then we might have a second sample, so that's sample 1, and then you might have sample 2 where you've got the same gene, but you've got a lot more reads. I'm not going to count this time, but say you've got 28 reads, so twice the number of reads. but none of my writing is transferring here, is it? Oh no, there it is, sort of. Okay, let's go back over that, see if we can get that to work. There we go. So lots more reads in this case. So say there's 28 reads there. But this library, when you go and you look at the total number of reads in this library, you'll find that you'll find that there's 24 million reads in that library. And what that means is that the difference between sample 1 and sample 2 here, the difference between 14 reads and 28 reads has nothing to do with gene expression and everything to do with the depth of the library that you've sequenced. So the fact that you've generated twice as many reads for sample 2 means that just on average you will get twice as many reads mapping to all of the genes in sample 2. So that's the thing that we've taken care of, quote unquote, by normalising, so by calculating the size factors, which is the first step that we undertook in DESeq2. And then the second step, which we haven't addressed yet, so step number 2 is to estimate what's called the dispersion. So dispersion is, it's not entirely true that it's another word. It's essentially another word for variance, although there's a relationship between dispersion and variance, but they're not exactly the same thing. But so this is the second step, is to estimate the dispersions, and the dispersion is a key parameter for the negative binomial distribution, which is the distribution that we use with RNA-Seq data to estimate whether genes are differentially expressed or not. This dispersion, which is what we're going to do in the next step, is estimate the dispersion of our count data, is it's a measure of the variance, and it's the sum of the biological variance, so that's the naturally occurring stochastic variance in gene expression, which we can estimate from our count data, but also what's called the shot noise. So the shot noise is the systematic experimental variation, essentially a measure of the uncertainty of read counting as a quantitative metric. So the dispersion is the sum of these two types of variance, the biological variance and the shot noise. For genes of low expression, for lowly expressed genes, the shot noise dominates. So the technical variation is the most important factor. For highly expressed genes, it's the biological noise that dominates. What DESeq does is it estimates this variance. So it's an estimate because you can't ever know the true variance without a huge number more measurements than we have here. And it estimates it in quite a clever way. So it doesn't just look at, in our case, we've got data sets where we've got 6 measurements for every gene because we've got 6 observations. And those observations fall into 2 categories, so we can use those categories for estimating a certain amount of biological noise. But that's not very many observations. Right? So if you're going to do an experiment to measure the average height or the difference in height between male and female students, for instance, this is the example I always use with my students. If you're going to do an experiment to measure the difference in height, then you're not just going to measure 3 male students and 3 female students, because intuitively in that experiment you know that sample size isn't large enough, and that you need to measure more students. And the more students you measure, the better estimate you're going to make of both the mean height and the variance of the height. And a perfect experiment is to measure all of the students at your university, and then be able to compare the male population to the female population with as much accuracy as possible, because you've measured all of the individuals in that population. But even in the case of measuring height, that's an experiment, generally speaking, that would be an experiment that would be too costly in terms of time and energy and all the rest of it. So what you do instead is you estimate the mean and the variance by measuring a sample of the population. And you take that, you make that sample as large as you possibly can. You're doing the same thing in a gene expression experiment. You're attempting to measure the mean of expression in different conditions, and you're doing that by measuring a subset of the population. In this case, three sets of cells in culture. Now, we know that 3 replicates in this condition in this circumstance isn't sufficient to measure the true mean and the true variance of that population. But we can, in gene expression experiments, we can benefit from the fact that we're not just measuring a single variable. So in the experiment when we're measuring height is, most of the time, height will be the only thing that you're measuring. You may also measure weight, and height and weight are clearly related, although not directly related. For instance, and then you're measuring 2 variables, and those 2 variables have a relationship, but that's not, you still a small number of variables, but you can measure those variables in a large number of people because it's not very costly to do that experiment. An RNA-Seq experiment is costly, both in terms of the time it takes to prepare the samples and sequence the samples, and also in purely monetary terms. So you don't take as many samples, you don't sample as many individuals, but we are measuring many, many more variables. And we can take advantage of that in an RNA-Seq experiment. So instead of measuring 1 or 2 variables, we've measured 60,000 variables because we've measured the expression of 60,000 genes. And there is relationships between the expression of those genes. They are not entirely independent measurements. And so what DESeq2 does, which is very clever, is it uses the variance across your whole experiment. So not just the 6 observations for a single gene, but the 6 observations for 60,000 genes to submit its dispersion estimates to what's called shrinkage. So it takes your measurements for one gene and it compares them to measurements for all of the other genes, and it borrows information about the variance from those other genes to shrink the estimate of variance for the gene that it's currently considering. It's called empirical Bayesian shrinkage, and it's a very clever mechanism for improving the robustness of these observations in experiments where you really don't, be blunt about it, where you don't have enough replicates. And there's always good reasons for why you don't have enough replicates in these experiments. But I mean, to be honest, unless you had thousands of replicates, it wouldn't be enough to do the experiment completely properly. So that's a clever thing that DESeq2 does. And that's the next thing that we're going to do. I'm just going to put step 3 in here as well. So step 3 is we apply statistics. And DESeq2 provides us with several ways of applying statistics. We're going to be deriving a statistic from a test called the WaldTest. So that's the statistics that we're going to apply. It's a statistic that's applied across the negative binomial distribution, which as we talked about previously, is a distribution by which we can model RNA-Seq counts. So, okay, so the next thing that we need to do then is step number 2, and that's estimate the dispersions. So, as with the calculate size factors that we did up here, estimate size factors, sorry, the way that we apply this is the same. We take our DESeq dataset and we provide it as an argument to a function which estimates the dispersions, and those dispersions get saved in our DESeq dataset. So we recycle the name of our DESeq dataset. So we do, let me shift this up the screen a bit. Okay, so we've got two datasets, remember, so we've got DDS1 and DDS2. So I'm going to do this with DDS1 first. The function is, so previously it was estimate size factors. This one is estimate dispersions. So estimate dispersions, and the one argument for estimate dispersions is our DESeq dataset. And so this function will, as the name suggests, estimate the dispersions for that dataset. And why has that not worked? Model matrix is not full rank, and that's because we subsetted. So we subset our dataset, but we didn't subset our col data, and that was probably a bad thing to do. So what we're going to do, okay, I know what to do. We're going to, right, so, sorry, I'm talking to myself. Okay, our running this estimate dispersions has failed. So we've got an error, and we've got an error because it's looked at the design of the experiment, and it's complained that the design of the experiment is not full rank. And the reason for that is that in our design we have 4 different levels. So we have mock infected and SARS-CoV-2 infected, and we have 2 different cell lines. So the combination of those 2 factors gives you 4 different levels in your design. And in this data set, only 2 of those levels are represented because it only contains one of the cell lines. So the model matrix is not full rank because 2 of those levels are just not represented in this data set. And so what we have to do is we're going to have to regenerate our col data. So what I'm going to do is regenerate the data set from scratch. So subsetting the data set superficially worked, but I've not subset it like that before. So superficially it worked, but in reality it didn't work because we didn't also subset the sample table when we passed it into col data. So what we need to do is we need to retrieve the counts for DDS1. We counts 1. I'm sorry about this. I didn't realize this was going to be a problem. Counts DDS1. Normalized is false, but that's the default. So that will give us our counts for that. And then we need to subset the sample table. Sample table 1 is sample table, and we want the first 6 rows. So that's 1 to 6 before the comma to do the subsetting. So you subset inside square brackets. And then we want the first 6 rows. And then we probably want to re-level the factor, but let's see if this works. And then we want to recreate DDS1 from that. And this time, instead of doing a DESeq dataset, oh no, we don't even want to do this. This is not the right way to do it. I'm retrenching here. Okay. We're going to go right back to the beginning. Sorry about this. I hadn't anticipated this, but this is me debugging on my feet. So, okay, so this is real bioinformatics, right? This is what happens when you're doing bioinformatics in real life, where you haven't had a chance to you've not run this before because you've not encountered this data before. And as I've said all the way along, I've not always done these things before because I don't always have time during the day to prepare. fully for that afternoon's lessons. So we've got an electrician in the house today, full disclosure, because our oven's broken. So I was worrying until about 3:45. I was worrying whether I'd have power today to actually do this. So I haven't prepared fully for today. I like to be fully honest with everybody. So I hadn't run this command before. I didn't know this command was going to fail. So we're now going to have to, now that it has failed, we have to debug it and we have to do it properly. So what I'm going to do then is we're going to go right back to the beginning. Not all the way back, but certainly all the way back to TX import. Because what I'm going to do is I'm going to cut the dataset in half at the point at which we've got our sample table. I'm going to cut the data in half at that point, and I'm going to let it bifurcate. So I'm going to do, at that point, I'm going to cut it into two, and follow through the analysis of experiment 1 and experiment 2. And so for the purposes of certainly today and probably early next week as well, I'm just going to do one of these experiments. I'm not going to try and do both side by side. Okay, so what I'm going to do is we've got our sample table, and our sample table actually is in perfectly good nick, but we want to pull our sample files back out. We've got our gene map, so let's copy that and let's put this in down here. So the above doesn't work, so split experiment before importing data, and that's what we're going to do here. So this is the input for TX import, if you remember. So TX import required this sample files vector, which is a named vector. So it has the path to the elements of the vector are the paths to my quant.sf files, and the names of the vector are the names of the samples. And actually, we don't necessarily need to rerun these lines. So let's comment this out, because again, we don't need. So if we go to code, you can comment the lines of code by picking the option from the code drop-down menu in RStudio, and that will comment the highlighted lines of code. So I don't need to rerun those, but I do need to subset that vector. So I need sample files 1 to 6. So the first 6 elements of it, and that's my first 6 samples. So that's the first thing I need to do. So we're going to do sample files. I can't remember what the cell line is. First sample table is NHBE, so let's call it NHBE equals that. Okay, so that's that bit, and then we're going to read the count data back in using TX import. So I'm going to copy that line and paste it in here. So we're going to call it count data NHBE. Is a TX import with sample files NHBE. So that will give us our 6 samples in from our count data. We then want to split our sample table. We want our sample, the first 6 rows of our sample table. So we're subsetting the sample table to give the first 6 lines of that. I'm going to check the factor, the conditions factor, and see if we need to re-level that factor as well, because we've removed 2 of the levels. What's next? Okay, so then we make our DESeq dataset here. Make our DESeq dataset like that, and then we So none of this is going to change the data that's inside these data sets. This data will be the same as the data that was in DDS1 before. So then it's that estimate size factors. But it will fix the design matrix, and we'll fix it properly. So if I'd have gone back and subsetted the counts back out of the DESeq data set, the problem with that would be that I wouldn't have got the lengths from the TX import object, so the normalization would have been different, and I want the normalization to be the same. So we're going to call this DDS NHBE, and we get it from count data NHBE sample table NHBE, and the design is conditions. And then when we estimate our size factors, we're going to recycle that vector name. So the last thing that I need to check before I run all this, this is fun. Okay, so that's the subset. And then that's the TX import, which should only read in 6 files, which it does. Okay, then we're going to subset our sample table. Let's have a quick look at that. NHB. There we go. So that is the 6 that we expect, with the 6 sample names that we expect. Let's check that conditions column. Yeah, so that still has 4 levels. So we need to change that column so that it only has 2 levels. So in order to do that, we do sample table $conditions factor. So it's mock. Let's just do mock and infected. We don't need to do the cell line anymore because the cell line and I want to do levels equals. Oh, hold on, let's get this right. I want to get the levels the right way around because you want your control level to be first. And by default, they're alphabetical. Okay, I'm getting there. Promise. Right, so rep each equals 2. No, 3. Like that. Okay, let's check this factor. See that it is what we want it to be. Yeah, so mock, 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 infected, 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 and the levels are not in alphabetical order, but they're in the order that I want them, with the reference condition first. So when you set up a factor for this kind of pairwise analysis, so here we've only got 2 conditions, so we're doing a pairwise analysis. The best way to put this into DESeq is with the reference level first of your 2 levels in your factor. So if this was in alphabetical order, it would be infected and then mock, which is the wrong way round for the analysis that you would normally do in DESeq2. So when I've set the factor here, I've told it the order I want the levels in, so that my reference level is first, and the mock being the reference here. So that's going to reset that that sample table so that our we don't have any additional levels in the factor that's being analysed. So it says it's dropped factor levels which had no samples anyway. Did I not run that? I did run that. Okay. Oh no, that's because NHBE. That's why. Okay. And then that. So it would have dropped them anyway. So from that we have learned that DESeq would have dropped them anyway. So you learn something new every day. So there you go. And now we're back to where we were before. Huh. A little bit of a diversion. Okay, so there we go. We're back where we were before. The normalized counts will still be the same as the normalized counts were in DDS before, but now we can apply the estimate dispersions method. So DDS NHBE equals estimate dispersions DDS NHBE. And so this calculates the gene-wise dispersion estimates for our data set and applies the shrinkage and gives us our final dispersion estimates. We can take a look at the results of this procedure. So there is a DESeq plotting function, which is plot dispersion estimates, plot dispersion estimates, which allows you to see the results of this procedure. And this is this is a useful quality control measure for RNA-Seq data, because we know what we know what we expect this plot to look like, and so we can look at this plot and see whether it conforms to our expectations. And this does look like I would expect a dispersion estimates plot to look. So there's a few things about this plot which is worth just drawing your attention to. So the first thing is something which I mentioned yesterday, which is heteroscedasticity. So mean and variance in RNA-Seq data are not independent from one another. So the variance is not continuous with the mean of the count data. So as your mean of normalized counts increases, so as you move from left to right across your x-axis on this plot, your variance goes down. So your dispersion reduces as your mean count goes up. And you absolutely expect to see that relationship. That is a normal relationship for RNA sequencing data. So the other thing that is worth noting here is, so what's been plotted here, the black points are the gene-wise estimates of dispersion. So that is for each gene, from the 6 observations for each gene, the dispersion has been estimated, and that raw estimation of dispersion is plotted on here as the black points. Then the mean dispersion with the mean, so average dispersion with average count is fitted. There's a relationship that's fitted, and that's the red points, is the fitted relationship between the mean of the counts and the mean of the dispersion, the average dispersion at that count level. And then the Bayesian shrinkage estimations are applied, and the black points are shrunk towards that red fitted dispersion estimate. And so the resulting dispersion estimates gene-wise are then the blue points that you can see on the dispersion plot. So that's your final estimate of dispersion for each of your genes. So you can see the results of the shrinkage on this plot. And so this looks essentially exactly how we would expect a dispersion plot to look. So that the relationship between the mean of the counts and the dispersion changes over the range of the counts, goes from high dispersion at low counts to low dispersion at high counts. And our observed dispersions are not too widely scattered around that fitted line. So there's not any crazy relationships going on here. You've not got sudden peaks in the dispersion at very high counts or weird deviations from that fitted distribution at any point. This is perfectly normal dispersion estimates plot. So this is a nice thing to see. It's a useful, another useful QC measure in this process. And then we've got step 3 that we talked about before, to apply statistics. So we've estimated the dispersions and we've had a look at it. And so now the final step, step 3. There are 2 groups of points. So you've got the blue points, which are scattered around this red line, and then you've got this second group of points which are set away from those fitted points, and these are actually blue circles around the black points. These are genes which are considered outliers in terms of their dispersion, and DESeq, if they're considered outliers, DESeq doesn't attempt to shrink their dispersion, so it's highlighting them as outliers. And that's what's gone on there. So that's why you've got this group of points which doesn't sit with this other group of points. So they've just been highlighted because they fall a certain distance away from the line of the fit line. So that's just another facet of this plot. So thanks for that question. That was quite helpful. Okay, so step 3 is to apply the stats, and the way that we do this is with, so we've got DDS NHBE, which is our data set. We're going to recycle that again, and to apply the statistics, we use the function from DESeq nbinom, so for negative binomial, WaldTest. That's the test that we're applying. And again, we put our DESeq dataset as the one argument to that function. Okay, so this is the final step of DESeq. So we'll run that. It'll take a few seconds to run because it's applying statistics to 60,000 observations. So it's doing a lot of statistical tests. And there we go, that has finished. Okay, so that is DESeq. And what, may you ask, have we actually done? Well, we can find out what we've done by extracting results from our DESeq dataset now, which is what we're going to do. Before we do that, the thing that I wanted to do was just highlight a shortcut for this process. So we've applied 3 different functions here from DESeq. We've applied estimate size factors here, done this one. We've applied estimate dispersions, and we've applied nbinomWaldTest. DESeq provides us with a shortcut. So the DESeq2 shortcut for those 3 functions is, we could have done this at the beginning. I'm not going to do it now because we've done those 3 things already. But there is a single function called DESeq, which applies all 3 of those functions. So we could have just applied that one function. But the first time that I run this through with people, I like to break it down into these 3 steps and explain them one by one. But when we do the second data set, maybe we'll just do this shortcut. Okay, I see questions, but I also see Peter fielding them very well. So I'm gonna let that continue for now. The final thing that I wanted to do today then was to extract some results from our DESeq dataset. So we've applied the statistics. DESeq knows what the differentially expressed genes are according to the test that we've applied. But we need to extract those results out. So in the same way as we had our counts function for pulling the counts out of the DESeq dataset, we have a results column function for pulling the results out of our DESeq dataset. So this is essentially an accessor method in the same way as the counts function was. So this gives us access to the results that are already stored in our DESeq dataset. So in order to apply this, you need to give it, actually, because we're doing a pairwise experiment, all we really need to give this is our data set. And this will generate results for us. And because we've set our experiment up as a pairwise experiment, so we've just got two conditions, and we've set the factor up in the right way round. So we've got the reference level as the first level of the factor. If we just ask for results, DESeq will give us the results for that one pairwise comparison that it's possible to do with that data. There are ways, if you've got a more complicated design, there are ways of extracting different sets of results out of the data set. But actually, for this data set, we don't need to know about those things. We could maybe look at a more complicated data set another time, just as an exercise to show you how that works. But for this simple pairwise experiment, our result table is available to us by just applying the results function to our DESeq dataset. Okay, so we're going to run this. There we go. So that's got us some results in our results table. So this gives you the results for every single gene in your experiment. You can get an idea of what's happened using the summary function. So we looked at summary before when we were introducing R. We looked at summary as a way of summarizing the variables in a vector. So the summary function for base R gives you the median, the mean, the interquartile range, and all sorts of other things for a vector, or for a matrix, or for the columns in a data frame. The people who wrote DESeq have also written a summary method for a DESeq dataset, or actually for a results table, which allows you to summarize what's in that result table in a nice, clear way. So if you run this summary function over your DESeq results table, you get back a little summary of what's happened. So there you go. This is what's happened. So if you remember, we had 60,000 rows, roughly speaking, 60,000 rows in our count table. So the first thing that DESeq2 does is it filters out all of the rows which have a total count of 0. So lots and lots of those genes we just won't observe in this experiment at all, not in any of the samples across the whole experiment, and they don't get observed. So those immediately get removed. And in this case, that leaves us with 28,000 genes to test for differential expression. Okay, so immediately we've lost the best part of 32,000 rows from this data set, because those genes just are not observed in this experiment at all. Their total count across these 6 samples is 0. Then DESeq also removes 2 sets of genes. The first set of genes that it removes are genes that are considered to be outliers. These are genes where at least one of the observations is too far away from the mean for it to be considered to be a real observation. So this is a quality measure, essentially. And it uses this metric called Cook's distance, and it applies a standard cutoff to Cook's distance for deciding which of the genes are outliers in this particular experiment. And so that, for this experiment, is excluding an additional 8 rows from our data set. And then the other thing that DESeq does is it applies independent filtering to this data. So of the remaining 28,000 or so rows, starting from the lowest total counts, so the lowest mean, the lowest number of counts in the row, it starts to filter out genes. So it does this because genes of low expression, so genes that have very low count numbers, are statistically much less likely to be differentially expressed. Those low counts are dominated by technical noise, not by biological noise. And it's very, very hard to accurately discern differential expression of genes of low expression. What DESeq does is it filters out the genes with the lowest expression across your experiment. And it does that in such a way as to maximize the number of genes that pass the statistical threshold. So what it wants to do is DESeq wants to preserve as many differentially expressed genes as possible. So if you increase this mean count filter, you will start to filter out genes that are genuinely differentially expressed, and you don't want to do that. But if you reduce the count filter, so if you filter fewer things, then your multiple testing correction gets more severe. We're going to talk about multiple testing correction. I think I'll go through the results table on Monday. We'll have a quick look at the results table now, but I'm not going to go through it in any detail. We'll talk about multiple testing correction then. But if you're less stringent with this mean count filter, so if your mean count filter is lower than, in this case, 8, then your multiple testing correction gets more punitive and you will lose positive results. So DESeq2 here is trying to balance finding all of the things that are genuinely differentially expressed while not filtering out some things that otherwise you might lose. And in this case, so this filter is dynamic, and in the case of this data set, it has filtered out all of the genes with a mean count less than 8. So the average normalized count across the whole data set, if it's less than 8, then that gene gets removed from consideration for the statistical tests. And that then filtered out an additional 12,500 rows, leaving us with only about 16,000 rows that we actually tested for differential expression here. So from a starting point of 60,000 genes, we have actually only tested about 16,000. But those 16,000 genes are the ones that are most likely to be differentially expressed. And then we have found that 301 of those are upregulated significantly with an adjusted p-value of less than 0.1. So this is perhaps not as stringent a filter as we might want to apply. And 170 of them are downregulated. Downregulated here means that they are lower in the non-reference condition. So the reference condition is the first level of your factor. So yes, in answer to the question, as Peter's just said, it will be on line 116 where I've set the levels so that the reference level of the factor is the first level. So the reference level is first, and the non-reference level is second. If you didn't set the levels in your factor, then by default there will be an alphabetical order. So infected would be first. So then your test, the base level for the test becomes the infected level, and so what you're then seeing as upregulation will be up in reference rather than up in infected. So if you've got them the other way around, then it will be because the levels are the wrong way around. Okay, so let's just take a quick peek at the results table, and then I will call it a day because it's nearly 5 o'clock, and we will take a look next week at the details of the results table and how to filter it for genes that are differentially expressed. So let's just take a peek at the first few rows of our results table. So head gives you the first 6 rows by default. The rows here are still ordered by just the row order, so they're ordered by ensemble gene ID. So it isn't a given that any of these things at the top of this table are going to be differentially expressed. We can quickly, if we want to, let's view it. So we'll use the view function of RStudio to open it. God damn it. It's not a data frame because everybody likes to do this differently. Sorry, there's been a lot of this today. I'm aware of that. Why is my R session now frozen as well? Let's just do as.data.frame. Let's just do it the quick way. There we go. That's better. Right. Okay. So, as I said, I will go through this table in more detail next week. But I just wanted to quickly, if we open it in view, then we can order it by ascending adjusted p-value, and we can see this is our the differentially expressed gene with the smallest p-value, and it has quite a robust fold change as well. Let's just take a quick look at that gene. Just because I don't want to leave you completely hanging until next week. Okay, so here we go. That gene is CXCL8. So I've just searched ensemble for the ensemble gene ID. So it's chemokine ligand. So yes, involved in some way in the immune response, presumably. So that sort of makes sense in terms of a result. So there you go. That's our top, in terms of p-value anyway, that's our top differentially expressed gene in this data set. And if you've got to the point of having differentially expressed genes, then hopefully you could reorder that. You can reorder that data frame by the different columns and find the one with the largest regulation and other things as well. We're going to look at this in more detail next week. Okay, so I'm not going to completely spoiler myself by going into any more detail now. What I am going to do, so I'm going to sign off now. It's the end of this week for me. What I am going to do is I'm going to upload this code right now before I step away from my computer. I'm going to upload this code onto GitHub, and I'll post a link in the notes under the video to point to the code, because I realised that in order to get it working, I had to write quite a lot of code quite quickly, and so I want everybody to be able to catch up with that. So I will post the link to GitHub straight away. I also want to draw attention to a survey that I've put online. So I've posted a link to this on Twitter, and I'm sure some of you will have seen it. But I've created a very short survey, basically asking how people are getting on, what people want to see next, and just asking for feedback generally. The more feedback I get, the happier I will be. I'm quite happy to act on feedback. I really do. I do want to know where people want to go next, because I'm enjoying doing this. I'm enjoying being here every day while I'm working from home. It's looking highly likely that I'll be working from home for a while yet. Because although lockdown in various places is starting to ease, it's not going to be immediate that my university is going to be going back to working from the office. So I'm still going to have afternoons where I want to do this, and so I'd like to know where everybody wants to go next. Okay, so look on Twitter for a link to that. I'll post a link again to that as well in the notes under the video. Okay, thank you everybody, and have a good weekend. If you are in the UK, have a good long weekend, and I will see you next week. So have a lovely weekend. Bye all. Keep safe, stay well. Bye.